Hello, my name is Grant Harrison. Today is August 7th, 2021, and I'm here to summarize a review article entitled The Genomic Code, a Pervasive Encoding Molding of Chromatin Structures and a Solution of the Non-Coding DNA Mystery. The DNA non-coding mystery refers to the function or purpose of the segments of DNA that do not code for the production of proteins, but rather just seem to take up space. However, a few recent discoveries have brought attention to the value that non-coding DNA may have within eukaryotic organisms. Bernardi's review essay outlines several key components or differentiations observed in non-coding segments. Bernardi describes the differences in two major types of groupings, or isocores as he refers to them. Type A being that of GC, pore domains displaying low heterogeneity are responsible for molding the lamina-associated domains, or the LADs, whereas type B is the GC-rich regions displaying high heterogeneity with single or multiple GC peaks that mold the topologically associated domains, or the TADs. Concerning GC pore region LADs, these chromatin fiber regions are stiffened and seemingly non-functional. However, as mentioned, it is likely that they provide the molding for which other sections of chromatin are to be bent against. The latter type of chromatin fiber, being that of GC-rich regions, has peak areas where GC nucleotides are almost exclusive to that region. In that region is where the fiber folds, allowing for cohesion to slide along the two bent halves, joining the opposite ends that are more AT-rich, also known as the CTCF binding sites. Barney's review seemed conclusive in establishing that although these segments may not code for specific proteins, they play a role in the organizational structure of the chromatin. As each type of segment is inhibitory to nucleosome binding, both the GC-rich and GC-poor segments, that a specific feature also reduces the amount of supercoiling that may be essential for the ability of chromatin when copying DNA from parent to daughter strands. The same feature may also serve as a role in shape retention of chromatin fiber, preserving its integrity and resilience. My personal opinion on the article is that although it details a breadth of previous research as a review article should, it failed to concisely provide any sort of short list in lay terms how non-coding regions have a specific function. The person not as astute in the field of genetics as the author may be left scratching their heads to find meaning in his interpretations. Lastly, there were several missing definitions and overly wordy explanations that were hard to follow, but after getting through it, one could kind of sort of deduce, make some deductions about how GC poor and GC rich segments of previously thought to be non-coding actually do serve a function within an organism.